In this video, we're going to look at the function of testes. Testes have two major functions: production of the hormone testosterone and production of sperm through the process called spermatogenesis. We'll first take a look at testosterone. Testosterone is produced by cells in the testes called interstitial cells, also known as Leydig cells. The release of testosterone is stimulated through a process of negative feedback. Starting at puberty, the hormone called gonadotropin-releasing hormone, also known as GnRH, is released from the hypothalamus. It travels to the pituitary, where it causes the release of the hormone LH, also known as luteinizing hormone. Luteinizing hormone travels to the testes, where it will stimulate the interstitial cells to produce testosterone. Once the testosterone levels reach a certain level, the hypothalamus can detect this, shutting off this whole process. Again, it's a nice example of negative feedback. Testes serve a lot of different functions in the male, but they're really important for growth and maintenance of organs. Many different cells in the body are going to react to testosterone, like bones, for example, and muscles. Testosterone is also very important for the male reproductive organs. It causes them to grow and mature and then be maintained later in life. Testosterone is also important for sperm production. It really enhances sperm production. Without testosterone, males are often infertile, despite the fact they can still make some sperm. Females also make testosterone. In the female, the testosterone is not produced by testes, but it's produced by the adrenal glands. And like males, it causes growth and maintenance of organs. Now we'll take a look at the process of spermatogenesis. Spermatogenesis is the production of sperm. The testes make the cellular portion of semen. Semen is the name for the fluid that leaves the male's body during ejaculation, and it's composed of both cells and fluid. So the testes are producing the cellular part of that. Spermatogenesis is going to involve a special type of cell division called meiosis. So we need to take a look at that process. But first, let's review mitosis and compare that to meiosis, because mitosis and meiosis are the two forms of cell division that occur in the human body. But they are for different purposes. Mitosis is for the purpose of making clones of cells. We need this for growth, and we also need this when we have to replace dead and dying cells. Meiosis, however, produces unique haploid cells. Haploid meaning containing half the amount of DNA. Diploid is the term for a typical human cell, and a typical human cell has two sets of every chromosome, so double the amount of DNA. Let's quickly review mitosis and then compare that to meiosis. As I just said, mitosis is for the purpose of making exact copies of cells. So you're going to start off with one cell, and at the end of mitosis, you end up with two cells. They're both identical to each other, and they're both identical to the cell that started this process. During mitosis, we take our starting cell, and we have to double the amount of DNA in the starting cell. Typical number of chromosomes in a starting cell is 46 in a human, so after we double that, we get 92. Then we take those 92 chromosomes and split them into two halves. 46 go to one side of the cell, and another 46 chromosomes go to the other side of the cell. Then the cell splits in half, so that you have two cells, each cell containing 46 chromosomes, identical to each other and identical to the starting cell. But during meiosis, we're going to make unique cells that are haploid, so we have to use a different type of process here. Like mitosis, though, we start off by taking our starting cell and duplicating the DNA. So we go from 46 chromosomes to 92 chromosomes. Something unique that happens in the beginning of meiosis is that these chromosomes are actually going to shuffle their genes around and basically generally rearrange themselves. And that's something that does not happen during mitosis. This is what will lead to the unique nature of each gamete. Now we have those 92 chromosomes, and we'll separate them evenly into two sets of 46. Just like in mitosis, the cell will then split into half, each half receiving 46 chromosomes. Now we're going to go through another process. So this is different than mitosis. Meiosis actually has two parts to it. Part one we just looked at, and now we're going to look at part two. During part two of meiosis, those two cells that we just made are going to split in half again. So at the end of the second part of meiosis, we have 4 cells, each with half the amount of DNA as the starting cell. So this is how we can get those haploid cells that are, of course, unique. During meiosis, we saw that there are 2 rounds of cell division, not just one, like in mitosis. We also saw that the amount of DNA in the ending cells is half haploid compared to the starting cell. And we also saw that we had this process of gene shuffling that went on, creating unique cells. There is a technical name for this process of genetic shuffling, and this is called recombination or genetic recombination. Chromosomes exchange genes, creating those unique combinations. As I mentioned before, meiosis has 2 parts. The technical names for these parts are meiosis 1 and meiosis 2. During meiosis 1, we start off with that starting cell, double the amount of DNA, get that genetic recombination to occur, and then split the DNA into halves and split the cell into halves. During meiosis 2, we take those 2 cells that we just made and we split the DNA in half and we get our haploid gametes. This is a pictorial representation of the process of meiosis, but it's simplified because it's showing a starting cell with 4 chromosomes rather than 46. So here you can see we start with 4 chromosomes, double that amount to 8 chromosomes. Here we can see that shuffling round of the genes, the genetic recombination, which is also known as crossing over, as seen here on the image. After the crossing over process is complete, the chromosomes are separated into 2 equal halves, and then that cell splits into half. So here now we have the 2 cells that are formed at the end of meiosis 1. These 2 cells then enter into meiosis 2, where they split their DNA in half and then split their cells in half, so that we end up with 4 cells, each haploid in nature, containing 2 chromosomes as compared to our starting cell, which had 4 chromosomes. This is an image of something called a karyotype. A karyotype is a picture of the chromosomes from a cell. So you can see here we have 2 chromosome number 1s, 2 chromosome number 2s, 2 chromosome number 3s, 2 chromosome number 21s, and then we have a whole bunch of X and Y chromosomes. Based on what you know about human genetics, is this a human karyotype we're looking at? If you said no, you're correct, because humans have 22 pairs of autosomes and then 1 set of sex chromosomes. This particular organism is lacking chromosome number 22, and the sex chromosome combination here is a little bit wacky, right? Typically in humans we have XX or XY. This happens to be the karyotype of a platypus, which happens to be my favorite animal. This is a human karyotype. 2 chromosome number 1s, 2 chromosome number 2s, 2 chromosome number 3s, 2 chromosome number 22s. This is that 23rd pair. Do you think this is a karyotype for a male or a female? What do you think? So we have to look at that set of sex chromosomes. If they're an actual pair, XX, that would be female. But if they're not a pair, then it would be XY. If you said this is a male, you're correct. Because in order for this to be a female, there have to be 2 of these big chromosomes, the 2 X chromosomes.